Harrison here. Today we're going to go over photosynthesis for AP Biology Unit 3. This is going to be a longer video, so if you need to move ahead, go ahead. Alright, so talking about photosynthesis, we're going to talk about autotrophs and heterotrophs. Autotrophs are your self-feeders. They're going to produce their own food. They cannot just go and chase down food. They have to be able to make their own food. And we're looking at that when we see plants. Heterotrophs are going to be ones that have to use other compounds by other living organisms in order to be able to metabolize. They're unable to produce their own food, so they have to go and find and hunt for their own food. Photosynthesis first evolved with what were called prokaryotes, and the first one we need to talk about is cyanobacteria. These are the early prokaryotes that help to oxygenate the Earth's atmosphere, and this produced oxygen for other organisms that eventually became like heterotrophs that had to have oxygen. Cellular respiration is going to help with that. So cyanobacteria is the first cell that started to photosynthesize and produce oxygen as a byproduct. These are going to be capable of photosynthesis, and these are going to be synthesizers that led the way for these eukaryotic photosynthesis organelles and cells. The site of photosynthesis happens in the chloroplast, and it's going to be found in leaves. This is going to be the area right here where you're going to find a majority of your chloroplast. This is the mesophyll. This is going to be the area where you can see these little green dots. These are my chlorophyll. Down here in the bottom I have some, but this is the area where light hits. And where light hits, I would expect to see a lot more chloroplasts or chlorophyll in this area. You also have what are called stomata, and stomata are the holes or the openings in the bottom of the leaf that allow a transfer of gases. So carbon dioxide can come in, oxygen can leave, water can go in. So this is the area here where I can have a transfer of gas. The structure of the chloroplast, this is a chloroplast. I have a double membrane. You can see I have two layers here. I have nucleoids. These are going to be the DNA that help to replicate the chloroplast. They're circular. I have stroma, which is the fluid that is inside this chloroplast. I have thylakoids, and I call the thylakoids, they're like little Oreo cookies, and they're going to lay on top of each other into these stacks. So each of these cookies, each of these little pieces is a thylakoid. If I have a stack of thylakoids, we call that a granum. So this is a stack of thylakoids. And inside the thylakoid, inside that Oreo cookie, is a space. And we call it the thylakoid lumen. So you think of like an Oreo cookie, that white stuff in the middle, that would be like the thylakoid lumen. That's the center of that cookie. So this is super important. When we start talking about like photosynthesis and where it happens here on this layer. In that little membrane right there around that red part, that's where photosynthesis happens, is inside that membrane. So we have a formula for photosynthesis. I have carbon dioxide and water with light make sugar and oxygen gas. So we have reactants. Reactants are what's going to go into the formula. And then I have products. This is what's coming out. So this has to go into the plant for the plant to be able to make sugar and oxygen. We got to have C's, H's, and O's for glucose. So this is kind of the big takeaway for photosynthesis. I have to make sugars. For me to make sugars, I need C's, H's, and O's. I have to harvest these to be able to make this sugar. The only place I can get these C's, H's, and O's is over here. I'm going to get my C's from carbon dioxide, I'm getting my H's from water, and I'm going to get some O's from my carbon dioxide over here. So I'm going to be able to get my C's, H's, and O's from over here for me to make this. So you need to think of photosynthesis as I am harvesting C's, H's, and O's in order to make glucose. I have extra oxygen left over from like water, and that extra oxygen is going to leave the plant. So this is why the plant has waste of oxygen. But that's the whole point of photosynthesis. I have to get C's, H's, and O's to make sugars. So we have what's called a redox reaction. These are when electrons are transferred from one reaction to another. And we have two that we have to talk about. One is oxidation. This is a loss of one or more electrons. Electrons are leaving, okay? I have a reduction. This is a gain of one or more electrons. What people want to say is if it's reducing, I'm losing. So if you have to think about it the opposite, that's fine. But what's happened, it's a reduction because it's gaining electrons from someplace else. It's reducing another atom, another compound, in order to get the gain on this side. So the best way to remember this, and this is how I remember it, Leo, the lion, says grr. Leo stands for lose electrons, you're oxidized. Gain electrons, reduced. So if you remember Leo the lion, he goes grr. If you remember that, that's going to help you. Lose electrons, oxidize. Gain electrons, reduce. So let's look at our formula again. I have carbon dioxide and water. What is happening to my carbon dioxide? Am I gaining or am I going to be losing over here on this other side? So I have a reduction. So over here, this is reducing. So this is gaining electrons. I'm gaining electrons from other carbons. I'm gaining electrons from oxygens. I'm gaining from hydrogens. I'm gaining more stuff over here on this side. Water is going to lose. Because see here, all I have are O's now. I've lost my H's. So I am losing electrons, so I'm going to become oxidized. So there are two stages to photosynthesis. We have a light dependent, I need light, and a light independent. I'm not necessarily needing light for that process to work. So with light dependent, I'm going to have sunlight and water come in. And out of that, I'm going to have oxygen gas. Over here, carbon dioxide is going to come in. And out of that, I'm going to get sugar. So my products going in is sunlight and water. And over here, I have carbon dioxide going in. So I'm going to have oxygen gas out, and sugar is going to be in. These are going to be my outputs. So let's talk about light energy for a second. Light, it affects the plant. Light has to go into the plant to start this process. So we look at this electromagnetic spectrum. These are photons of light. So light comes in different waves. So when we see red as a very flat, very wavy line. So when it hits our eyes, we see that color as red. When the line gets more jagged, we start seeing more blues and purples. So this is a more jagged wavelength. This is a more flat, longer wavelength. So when we see colors, this is what we're seeing. This also gets certain pigments excited based on the wavelength that hits it. Now, the way we see color is an object is going to have some of these wavelengths in it. If the color does not, that wavelength's not in that object, that color doesn't get absorbed. It gets bounced out. So this blue paper, I have red pigment in here. I have orange and yellow and green and violet. All those pigments are in here. The pigment color that is not, or the wavelength that is not being absorbed is blue. So that blue gets bounced out. These other wavelengths get absorbed. Here I have orange, yellow, green, blue, violet. These wavelengths are in this paper, so they get absorbed in. This red wavelength is not, so it gets bounced out. When I have something that is white, all the colors are not in there, so all the colors get bounced out. When I have a black object, all the colors are absorbed. So when we look at plants, plants have to have a certain wavelength of light. If you've ever had a plant in a house and it's not by a window, the lights in your house will not allow it to grow. It's not hitting the right wavelength. So if you have plants inside of a house and you want them to grow, you have to go to the store and you have to get special light bulbs that are going to give you this wavelength of light and is going to benefit and start to excite these pigments that are inside the leaves. So we have three different types of chlorophyll, or different types of pigments. We have chlorophyll A, which is this one here. This is your primary pigment. This is going to be part of that light reaction, and it's going to involve blue-green light. Okay, I'm going to have some blue-green light here. But you can see, I can also absorb some of these oranges and some of this red with chlorophyll A, but I'm going to absorb a lot more with this color. Chlorophyll B is an accessory, which means it's going to be a helper, and it's going to involve some yellow and green light. I can come in here and get some yellow and green and some over here also. I also have carotenoids, which is this line here. This line here is going to help broaden the spectrum, help bring in more light waves to help excite pigments. It's going to involve yellow and orange. This is what you see whenever you go and you have bell peppers and you see red and orange bell peppers. What you're seeing is the carotenoid. You're seeing that yellow or reddish color. And what that does is it helps protect the plant from UV rays. So the plant doesn't have, it's not, the UV rays isn't damaging the cells inside the plant. So this provides what's called photoprotection, and that helps produce these pigments that help to protect the cell, and it keeps it from damaging the chlorophyll. And when you look here, you see this part here, the green. This part here, the plant doesn't absorb this light, so I'm not going to get a lot of interaction, a lot of production stimulating the pigments with this wavelength of light. So we have the light dependent reaction, and remember, water and sunlight have to come in. And then I'm going to have a net product. So this is all the stuff that's going to come out of this reaction. We already know I'm going to get oxygen. That's going to be a waste product. The plant's going to get rid of that. But I'm also making things in this cycle that the plant needs. So the plant needs ATP, and I need what is called NADPH. This needs to go to this cycle over here. So it happens in the thylakoid membrane. Remember the little Oreo cookie, the little outside of that cookie is the membrane. This is where this is going to happen. And then it's going to convert solar energy into chemical energy. I'm taking sunlight energy and I'm turning it into chemical energy. So first thing that's going to happen, light energy is going to be absorbed by photosystems, which are these pieces here, this PS2, PS1. And it's going to drive the movement of electrons from water, which is going to come here, to form NADPH. I'm going to have NADPH form here. So photosystems 2, photosystems 1. And you notice photosystem 2 comes first, and then photosystem 1. Photosystem 1 was discovered first. And then later on they recognized that this photosystem started the process. Instead of renaming this, they kept it. So photosystem 2 comes first, and then it goes to photosystem 1. All right, so what happens is light energy hits the photosystems. Inside these photosystems are pigments, and they're going to get excited by this light. Water's going to come in, and it's going to pull off an electron. I'm going to have electrons get pulled off this water. The electron is now going to get pushed into the PS2. It's going to get excited, and it's going to start to move through this trail. This is called the electron transport chain, the ETC. This electron is going to keep moving through. It's going to come to PS1. It's going to get re-excited, and it's going to jump out, and then I'm going to attach to NADP+. Hydrogen is going to attach to it also, and then I'm going to have NADPH. So this is a product that is going to come out of this light-dependent cycle. So water comes in, electrons are going to move through the PS2, through this electron transport chain, the ETC. It's going to come to the PS1. Electrons are going to move through. They're going to attach to this NADP+. Hydrogen is going to attach to it also, and I'm going to have NADPH. So this is going to be carriers of electrons. So NADPH, I'm going to take one hydrogen proton and 2 electrons. So these electrons are coming out of that electron transport chain. And you need to think of NADPH like a taxi. Here I have NADP+. See that the H isn't there yet? So if that H isn't there and I'm positively charged, That means I have no hydrogen, I have no electrons. So once this comes together and attaches to this NADP+, now I have NADPH. So now this is showing the hydrogens attached, and I have my electrons attached to it also. This is now going to leave that light dependent cycle, and it's going to head off towards the Calvin cycle. Step 2, water is going to be split and oxygen gas is released. So here my electrons moved through and I made NADPH. But now when this water comes out, I rip off these electrons, this water is going to split. I'm going to have oxygen gas and hydrogen protons are going to start filling this space here. This oxygen gas is then going to leave, okay, and then I'm going to have hydrogen protons build in this membrane. What happens now is I need to make ATP. ATP now needs to leave this area. I have to make ATP. So we call this chemiosmosis. What this is what's called ATP synthase. And ATP, if you've ever seen the top of a house and you see like the little whirly birds, the heat comes up from the roof and it causes these little fans to move. This is what this is like, and it's going to spin. And what happens is I have a ton of hydrogens here. I have a bunch of hydrogens. And if you remember, when I have a high concentration of substances, it wants to go to a low concentration. So I have a high concentration, and the only way these hydrogens can leave this high concentration is to come here and to move through ATP synthase. And it's just going to start running through ATP synthase. When it does, it hits the sides and causes it to spin. As it starts to spin, this ADP is going to grab a phosphate, and the I means inorganic phosphate, it's just a phosphate, and that phosphate is going to get slapped back onto this ADP and turn it into ATP. The only way that this can happen is due to this concentration gradient of hydrogens coming through. So I have ADP and a phosphate make ATP because of these hydrogens coming through, creating this spinning motion that creates enough energy for this to be attached back onto the ADP. So here I have a photosystem. This is going to be the little photosystem 2, photosystem 1. And these little green dots here, these are my pigments. The light comes in and gets these little pigments excited. And they start to vibrate and they start to move. And this energy gets passed through these pigments. You have a special pair of chlorophyll, and this chlorophyll A is here. And we have different strengths. We have a 680 and a 700. And this chlorophyll 680 or 700 is going to get really super excited. And an electron from that water is going to be placed here. And then this is going to get so excited, that electron is going to get so excited that it's going to jump up to this. This is my reaction center complex. And my electron is going to go here, and that's going to give it enough boost or energy to slide down that ETC to go to the next area. So let's look at this one. So here's my P680. This is my chlorophyll A. I'm going to have light come in. And then all of a sudden I'm going to start having this water come in. And then these little pigments are going to get super excited. My electron is going to get moved in. I'm going to have one oxygen molecule and 2 hydrogen molecules are going to come off. So this is half an oxygen gas. Another water molecule will come in, and then that oxygen will come on, and then I'll have 2 oxygens, and I'll have oxygen gas. So right now I have half of an oxygen gas and 2 hydrogen molecules. This electron got pulled off and put here in chlorophyll A in this one spot. So now these pigments are going to get super excited. When they get super excited, they're going to get this electron really excited too, and it's going to jump up here to this primary acceptor area. So it jumps up, and then it's going to move through this ETC. This is my electron transport chain. When it gets here, it's lost energy. These electrons have come through, and it's lost energy. It has to get recharged. And the only way it can get recharged is again through these pigments. So photosystem 1 is now going to recharge this electron. So I'm going to go through, get all these super excited. It's going to take this electron, jump it up to this primary acceptor. And then it's going to have enough energy now to be able to go down, come down here. It's going to attach to NADP+. A hydrogen's also going to attach to it. And then the electron's going to go here. And this NADPH is going to be ready to go to the other part of photosynthesis. So all of that happens here. Okay, so here's my photosystem 2, my electron transport chain, photosystem 1, the end of my chain, I take NADP plus hydrogen, and I turn it into NADPH. These electrons get moved on here. I have my water comes in. This is where my electrons got ripped off. My water is going to break apart. I'm going to have one oxygen come off. 2 hydrogens are now going to come into this space. And then when it happens again, I'll have 2 for make oxygen gas. This oxygen gas will leave. But remember, we talked about these hydrogens. These hydrogens have to build up. I have to run ATP synthase. But just having these hydrogens is not enough. I've got to bring more in. So what happens is I'm going to have hydrogen protons outside in the stroma. Remember that gel-like substance outside of those Oreo cookies? This is that Oreo cookie. And then this is the outside of it, and this is the inside of that Oreo cookie. Remember, there's stacks of these. In this membrane, I'm going to have my electrons move, and they're going to move through this chain. And remember, electrons are negatively charged, and anything that is negative is going to attract positively charged particles. So what this electron transport chain does is it brings in positively charged hydrogens to help add more hydrogen protons to the center so I can build up a huge concentration. So they just want to fly through this ATP synthase so I can make more ATP. So these hydrogens are going to get pumped through. As these electrons go through, it's going to allow these hydrogens to come in. And then I'm going to have a huge buildup of these hydrogen protons. These hydrogen protons they're going to start flying through this ATP synthase. And now I'm going to have hydrogens out here. I'm going to have ADP plus phosphate. These are going to get slapped together inside of this ATP synthase. And then I have ATP. So this whole process to make ATP is due to this concentration gradient of hydrogen protons I have built inside this thylakoid lumen. And I build it because I'm pulling off hydrogens from water and I'm pumping hydrogens in through these electrons. All right, we call this chemiosmosis. So chemiosmosis is when I'm able to make ATP due to a concentration gradient, due to like a hydrogen, it's like a hydrogen pump is what's coming through. So that concentration gradient from great to small. And then these hydrogens, when they come out, they're just going to get pumped back through. These ETCs are just going to kind of keep cycling back and forth. Some will get tagged onto NADP+. So this is just going to kind of keep moving hydrogen protons around. All right, that's the first half. We just talked about what happens here. And if you need to pause for a second, take a break, come back, that's fine. Like, this is a big process. So let's look at this. So I had water come in. This is what pulled off my electrons. I pulled off hydrogens to fuel that hydrogen pump. And I have oxygen gas as a waste. This was all fueled by the light, getting those pigments excited. This process gave me ATP and NADPH. I need this for the next cycle. So the Calvin cycle occurs in the stroma. This is the gel-like substance outside. NADPH, ATP, and carbon dioxide now have to come in. So carbon dioxide from the air is going to come in. What happens is this. Okay, this is kind of a process, so if you need to watch this a few times, that's fine. I'm going to bring in, for right now, 3 carbon dioxides. I can only pull in 1 carbon dioxide at a time, but I need 3 for this first process to work. So one of these 3 is going to come in, and that carbon dioxide is now going to bond with what is called rubisco. Rubisco is this part right here. It's a 5 carbon sugar. 1, 2, 3, 4, 5. And then I'm going to add one of these carbons to this group. This oxygen is still here. We don't show it because it just gets too messy. But these oxygens are still here. Okay. But we're going to look at it like this. So another carbon dioxide is going to come in, and I'm going to make another 6 carbon molecule. I'm going to bring in another one, and I'm going to make another 6 carbon molecule. So now I have 3 molecules, 6 carbons, phosphates on both ends. But this is very unstable. It does not like to be at 6. So it breaks apart. So now I have those 3 carbons here, 3 carbons here, and now I have 6 of these molecules. We call this 3-GPA. So I have I have PGA. These are PGAs, 3 PGAs. I have 6 of them. We call this carbon fixation. I fixed carbon to that rubisco. I'm fixing it to that molecule. So we call this the carbon fixation. I'm bringing in carbon and I'm fixing it to the molecule. So now what happens is I'm going to have to bring in NADPH. Remember, I have to make sugar. I have to bring in C's, H's, and O's. I just brought in my C's. Now I'm bringing in my H's, or I should say I brought my C's and my O's. Okay, remember my O's are on here too. I bring my C's, my O's, and my H's. I need to put together my sugars. So remember those NADPHs we made from the light dependent cycle? Those are coming here to the Calvin. So this NADPH is going to start pulling off these hydrogens. But remember, this can't happen on its own. Something has to make it come off and pop off. So that's when we have what is called ATP. ATP is going to come in, and it is going to start ripping off these hydrogens and then ripping off some of these electrons, and it's going to start attaching them to these PGAs. This NADP plus is now going to leave, and it's going to go back to the light dependent cycle. This ATP has to use a phosphate to get this endergonic reaction to start to work. Remember, endergonic, it doesn't want to, so this has to start that reaction to get it going. So now I have ADP. So I have NADP plus has lost its hydrogens, has picked them off and put them here. Now it's empty. This taxi has lost its carriers. It has to go back. The taxi is going to drive back to the light dependent cycle, pick up some more H's and electrons. This ADP is now going to go, and it's going to go back to the light dependent cycle. It's going to pick up another phosphate, and it's going to go through the ATP synthase to be made back into ATP again. So I'm going to have 6 NADPHs and 6 ATPs to make this change into what we call G3P. We call this a reduction because remember, Leo the lion says grr. When you lose electrons, you oxidize. You gain electrons, you reduce. So I am gaining electrons here, so I am reducing 3-PGA to G3P. So now I'm going to look at these molecules here. So now what happens, one of those is going to get pulled out. One of these 3 carbon molecules is getting pulled out. These other 5 are going to be used to make rubisco. This will be used to make glucose. So right now I've only made half a glucose molecule. So people are like, wow, this is like crazy. Like I had to bring in 3 carbon dioxides and do all this just to make half a glucose molecule. Making stuff's not easy, okay? So this is that process. So this is just half a glucose molecule with just 3 carbon dioxide. So 3 turns of this is kind of what we're getting at. So now I take these 5 pieces, and these 5 pieces are now going to regenerate. I have to make Rubisco again. So what happens is, remember, Rubisco is the 5 carbon sugar. So I've got to take these pieces and I have to rearrange them back into 5 molecules. So if you look, I've got 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So I can make 3 Rubiscos with what is left over here. But I can't do it on its own. It's just not naturally going to do that. So ATP has to come in and give it that boost to be able to reattach it. I'll have another phosphate come in, another phosphate will attach to that. So these are going to come together. So now my Rubisco has been regenerated. It has been remade for me to allow the next carbon dioxide molecules to come in so I can do this process all over again. So carbon fixation, I'm fixing the carbons to the Rubisco. It does not like to be a 6 carbon molecule, so it breaks. NADPH is going to come in, is going to put in the H's. This NADP plus has been depleted. The taxi is empty. It's going to go back to the light dependent cycle and pick up some more H's and electrons. ATP is going to help this process happen, and as it does, ADP has to leave and it's going to go back to the light cycle to be made back into ATP. This 3-PGA is going to be reduced. Electrons are being added to it. Out of those 6, 1 will be pulled out. The other 5 will be regenerated back into Rubisco with the help of ATP. ATP is going to give it the energy it needs to reorganize into 3 5 carbon molecules. So the second set of carbon is now going to give me enough to make 1 glucose molecule. All right, so I take a glucose from here, I take my glucose from here, and now I have 1 glucose molecule. So they say you have to have 6 turns of this. I had to bring in 1 carbon dioxide, 2 carbon dioxides, 3 carbon dioxides, and then I had to do it again. 4, 5, 6. So I have to do this 6 times for me to make 1 glucose molecule. So here's an important slide, and this is showing you all of your inputs and your outputs. My light dependent cycle, this is the one where the light comes in. I bring in water, ADP, and NADP+. Remember, this just got depleted from the Calvin cycle when I was making my sugars. This has to go back and get replenished. My outputs is going to be oxygen gas from the water. ATP from chemiosmosis is going to go through that concentration gradient, go through ATP synthase. ATP is going to get made, and then NADPH is going to get put back together. This is my ETC, my electron transport chain. I'm going to have hydrogens put on. I'm going to have electrons attached to this NADPH. These are now going to go back here to this cycle, the Calvin cycle or my light independent cycle. I'm going to have 3 carbon dioxides come in. I'm going to have 9 ATPs. I'm going to have 6 NADPHs. So remember, 6 of these 9 are going to help attach the NADPHs. The other 3 are going to be to attach the Rubisco back together. And remember, this is just half a glucose molecule. Okay, out of that, I'm going to make 1 G3P. This is half that glucose molecule. I'm going to make 9 ADP, which are going to come back here. And I'm going to make 6 NADP+, which are going to go back here. So I am swapping these pieces back and forth. Is kind of what I'm doing with those. So carbon fixation, this is an evolution trait that's happened in plants. Plants have what's called a stomata. And the stomata are these little vesicles that are full of fluid, and they can swell and open up. When the plant needs to open up the stomata, it's going to relax and open up, and it's going to allow gases to come back and forth. Water can escape, a little bit of water can come back in. And so this is going to allow carbon dioxide to come in and oxygen gas, remember the waste, to be able to leave. Water can also leave through the plant through these stomata too. It's also why when you go to a greenhouse, it's kind of humid inside. These stomatas can open up and allow water out. So carbon dioxide in, oxygen gas out. I can also lose some water with that too. So this is a problem. So what we call C3 plants, these are just regular plants, they can have an issue. So the first problem is a hot day. These C3 plants, they're C3 because they're PGAs. Remember, I got those 3. I had 6, they broke apart. So 3 carbons, that's why they're called a C3. This C3 plant has to conserve water. It does not want to keep that stomata open, but I have to have stomata open to get carbon dioxide and to get rid of my waste. So they close their stomata during the day to conserve water. I don't want to get rid of all this water. But closed stomata means I have no carbon dioxide uptake. And no carbon dioxide uptake means I have less sugar production. I'm not able to feed the plant. Another problem is that when I have my stomatas closed, I cannot bring in carbon dioxide to fix to the rubisco. Remember, carbon fixation. I need to fix that carbon to that rubisco. What happens is oxygen ends up binding to it instead of carbon dioxide. And then this causes oxidation of rubisco. Remember, Leo the lion says grr, lose electrons, you oxidize. So rubisco is losing electrons, it's losing material. So that means it's going to break down. And we call this photorespiration. And when you have photorespiration, it can break down as much as 50% of the carbon fixation. Like you're really starting to damage the plant. So this becomes a big issue for C3 plants. So they have an evolutionary trait that some plants have been able to use to help overcome this dilemma, this problem. And one group are called the C4 pathway. Here they have separated areas inside the chloroplast. You have the area where you do your Calvin cycle, but you have a separate area where it actually fixes carbon dioxide to another molecule. So you have an enzyme and you have a PEP compound that comes in. This PEP carboxylase, remember ASE is an enzyme, it pulls this carbon dioxide and attaches it to this PEP. It turns into oxaloacetate and it turns into malate. And this is carbon dioxide is just tagged onto here. Later on, when the plant needs it, it's able to take this molecule that has the carbon dioxide fixed to it, pulls it in, and is able to pull that carbon dioxide off that, and rubisco is able to work it. This malate then loses this carbon dioxide and turns into pyruvate. And then ATP comes along, helps rearrange it back into this, and is able to store carbon dioxide into these little boxes to be used later on. Examples of this is going to be corn, grass, sugarcane. This is going to be what these type of plants can do to keep able to not lose a lot of water during these hot summer days. Another one is called a CAM pathway. These only open up at night. Stomatas only open up at night. But instead of being separate areas, that's actually stored in vacuoles. So you have this, again, the PEP. I have my PEP carboxylase comes in, carbon dioxide fixes, and then it turns into oxaloacetate. I will have malate or some other organic molecule will come in, but this carbon dioxide is tagged or in that little box that it makes. Then during the day, so at night it's open, it's allowing all this stuff to happen, and then during the day, when you have the light dependent cycle working, it's going to start making all this stuff. And then when it needs to make sugars, It's going to bring in these organic molecules. It's going to pull off the carbon dioxide, and then Rubisco is going to be able to do its job and make its sugars. This material will then lose the carbon dioxide, turn back into a PEP, and it's going to keep doing this cycle. So for at night, this process is kind of coming in. Carbon dioxide is fixing it, putting it into little boxes. Think of it like little storage containers to be able to be used during the day whenever the plant is able to make more ATP and NADPH. All of that stuff happens during the daytime. Plants that do this is going to be pineapples, cactus, succulents, jade. Those are going to be ones that are going to just open up at night. All right, that's a lot. It's a lot going on with photosynthesis. It's probably something you're going to have to watch a couple times. I would recommend drawing it out. I would recommend talking about it. I would recommend trying to tell somebody about it. The best way to understand this process is to kind of visualize and do it. So if you guys have any questions, you let me know. We'll talk to you later. Bye.